Thank you, thank you. I know I'm the last one before lunch, so everyone's hungry, but this is fun. We got videos. And as you can see, we have a little tiny one here who, if you've seen him around, is a lot bigger now, right? So I really appreciate many of you have gotten to meet me over the past day and a half, and some more will meet me today and tomorrow. So if any of this is striking your fancy, or you just want to talk about the brain, I'm just going to be hanging out upstairs at any time. So if you need a break from downstairs and want to just come talk to me, please do. I also really have to emphasize that this is a partnership with Dr. Nicolò Pini, who's at Columbia University. We came together for that meeting in 2022. We're dear friends and colleagues and excited to kind of really propel what we're doing here. So I have more slides than time, so I have a timer and we might have to skip a couple things, but I'll give you the big kind of picture. But I really want to emphasize the fact that this is an important meeting because we have people across all of these different levels of who we are as humans, right? So we're really here to kind of think through, what's happening in MED13L as a genotype, but it needs to connect to each of these different levels. And this is important in particular if we're thinking of not necessarily just pharmacological interventions, but what things are going to really help our kids and people find that most best life that they can. And we spend in my lab a lot of time thinking more from the top down, right? So we think about these clinical diagnoses. I work a lot with autistic kids, but we also know that there's other kind of clinical diagnoses that will be linking back down to the genotype. And so what we end up doing is creating what we call biomarkers. So we're using the brain to try to understand between what we observe in our people back down to the gene itself. So if you've heard BioGene, it actually is an acronym. I was kind of impressed with myself for this. And we have a website. There's lots of videos on here. And we have social stories. We just made new ones that we need to post to the MED13L group because we had some inspiration from kiddos that love dinosaurs. So we have some brand new social stories. I've been doing this for many, many years, and I recognize the fact that particularly our rare groups are not going to come to me wherever I am. So we tried our best to go to them. So I'm known for taking the EEG in my car and taking a 3-month road trip around the world to see everyone. I love coming to these meetings because you're all here in one spot. So if I look tired, that's because we're planning to do EEGs with over 60 people in 3.5 days. So we are tired. And we have 3 different primary tasks that we've been using. And I want to just kind of emphasize what these are and why we're having your kids do these different EEG tasks, because we're not researchers that are specifically looking for epilepsy or sleep disruptions. What we really wanted to know is the everyday brain. So how are our brains being built and able to understand information in the world? And these 3 different tasks help us to get there. So a lot of, you'll see some pictures and videos from other gene groups that I've worked with, and we're going to continue to grow, but I think after this meeting we are going to have, well, I won't spoil that just yet. There's a slide with the number, but we'll really be pushing forward on what's happening with MED13L in the next coming months. So for those of you who haven't gotten to see what this looks like, this is not the gel-based EEG you might do at the hospital where you have to glue every single electrode and wrap your child and have to constrain them and wear a backpack for 24 hours. We use this flexible net. It stretches, it's wet, so we do that instead of using gel, but it goes on in just a matter of seconds. So here's just a little video from my dissertation way back in the day, just to showcase the fact that we can put the hat on and usually we can redirect, distract, and be watching iPads and different things. And then we're collecting brain data in a matter of 20 seconds, which I think is really, really cool. So this is one of our hype videos for a different gene group, but I think it gives you a sense of what it takes for us to kind of do this work. So I can put everything down into just 2 big suitcases. And this is me practicing in my friend's basement on how to set it up as quickly as possible. And then I think you get a really nice snapshot of the variety of kids that we see. We have a lot of folks that are needing supportive seating. We have kids that fall asleep. we have siblings that are super curious and become really great helpers. I teach them how they can help me put a little bit more water on the hat. And this is something that we can do with kids of all types. So regardless of what language they have, regardless of their mobility level, this is something that really everyone can accomplish, because everyone has a brain and that brain is working and understanding the world in different ways. And that's what we really want to be decoding. So we had two EEG testing rooms with Nicolò Pini as well, sponsored in part by Simon's Foundation at the Baltimore meeting. So that was 3 years ago. And we saw 43 families overall. And you can see here that we ended up seeing 14 MED13L groups. And I will say, you guys are interesting, because I think it was Katie or somebody gave a rousing speech, and then all of a sudden you all came to our table and we were slotting you in. I was making up spaces. I think I was eating a sandwich under the table. It was wild, and we got as many as we could. And that was super, super cool. I also have some other, two families also participated when I was a postdoc in something called the Tiger Study at the University of Washington. And this weekend, we've already seen 16. I think we're scheduled to see closer to 25, 26 families if we can get it all to work. And I know we're running out of slots. So if you try to sign up for, I think we only have slots tomorrow, but you can't get one and are still interested, come find me, because we'll figure this out. Come bombard me, we'll find those slots. And the important thing for us is that it's not just new families. We really want to understand how the brain is developing and changing, and how that helps our kids navigate the world, right? And how those brain processes are being built to support, and whether or not things that you're doing on your daily life are helping, right? So is it that you just started speech therapy and now we see a boon? So these are some of the things that we're trying to track, and why we want to see kids as often as we can. All right, so I want to shift a little bit and talk about one of these specific kind of markers that I'm really interested in, and in part because I think after lunch there's a session on more the basic science side. And this is a really nice example, I think, of how what we see in our human kiddos and people also could be mapped back to our animals. So we've been focusing on attention, and specifically what we call attention habituation, which is kind of like how attention is changing in real time. And this is something that we can measure in our kids. We can measure, as you'll see, in flies or the mice or the zebrafish. So this is something that we really can put into a model, and the models are something that we can then test out from drug potential interventions and things that we can do at the big level that we can't do with our kids necessarily. And it's a really important thing for us in life, right? So you all are paying attention to me, so gold stars for the day, right? But something, you might hear a sound, or you might see someone make a sudden movement. There's things that grab our attention over time. So what we tend to focus on is how new thing that just appears captures your attention, and we call that an orienting effect. So if I gave you, some of you have seen this before, but if I gave you a picture that looks like this, you might be looking at this picture and suddenly you might see this pop out, that one of these things is not quite like the other. And what's important is that our attention system is pretty smart. So whether or not you know it, your brain is starting to look through this information and decide, well, is this redundant information? Is it important? And as we are given new examples, we become a lot faster at kind of finding the rule or finding the thing that's a little bit different. So kind of finding that pop out in this kind of visual task. So what we do, instead of using vision, because not all of our kids are going to focus on a screen for 8 minutes, I have no control over where they want to look, and I don't really want to force them. So what we do is we let them watch whatever they want, we make the video be muted, and then we play a series of tones for them. And some of you parents, I've seen you in these rooms, you know that's not the most fun thing to just listen to these annoying beeps for 8 minutes. But what it happens is that you have these kind of moments where you hear this thing over and over and over again, and that attention part of your brain is starting to say, okay, that's information I don't really need. I can kind of stop paying attention as closely. But then you have this new sound that pops out, and then a part of your attention system is like, that was important. And then we have these really weird sounds and chirps and chimes and squelches, and those capture a whole other part of our brain. And I don't think we put audio on, which is totally fine, but this is an example of a kiddo listening and watching. And I wanted to just kind of emphasize the fact that you see that we let them sit. We're not like strapping their heads to anything. We know that they're gonna move and potentially scratch the net. That's partly why this is 8 minutes, because we can have sections of data that are maybe like artificial with movement, but we have plenty of time, and they actually kind of like some of the sounds sometimes. Okay, so what do we actually measure? We're looking at what we call this auditory response. And what I know from autism is that we see this sticky signal where we have that, particularly when that new sound pops out, we have a bigger response in this thing called the P3A. And this is what the P3A looks like from the MED13L kids that we had before this week. And you can kind of see in this red arrow, that's like the effect that we're looking at. That's the pop-out effect of that new information. But we can also pull out other things like this N1 effect, which is just more of a detection. So that's more of an automatic feature. And then this P3 is more about like, how do we make sense of that information? So, something's different. Now what do I do with that information? So looking a little bit more at what we see in our MED13L groups, I'm showing you here in these plots, a bigger attention effect is it's gonna be higher on the screen. And for both our detection and our attention orienting effects, we're seeing smaller attention effects with our MED13L individuals. So this is just the percentage of kids that are showing a positive value here. So overall, and this is related to kind of idiopathic autism and neurotypical development as well. But it's really important that we keep seeing kids, we see kids at different ages. So you can kind of see this distribution. This attention system is changing dynamically as kids get older. So one of the things that we're also starting to do is look, well, how are these attention effects changing with age? And one of the things I just kind of want to quickly kind of show, and I'm just trying to emphasize here by the size of these arrows, is that particularly for this red response, which is that orienting response, we actually see a very neurotypical response. But I know I'm not supposed to move, yeah. So you see how their red line is increasing. So as our MED13L individuals are getting older, we're seeing that there's more of this heightened response to those things. Whereas in neurotypical development, it's more that they're not caring as much about that repeated thing over and over again. So these are different ways in which they are attending and tell us something really, really classic about the brain. So I'm basically out of time. So I'm going to kind of give you like some punchlines in the next 2 minutes. One other thing that we kind of look at is kind of how, again, this changes dynamically within the 8 minutes that you're listening to these sounds. That was my own timer to wrap up. So we see sticky attention in autism. So what do we see in MED13L? Well, actually, we see some very similar trends. So in the top one, you can kind of see that MED13L and neurotypical development are both kind of showing some initial, what I'm calling increases. I'm happy to kind of walk this through later. But when we look at that red response, it's pretty flat. So what this is telling me is that the MED13L brain is staying pretty stable to these same sounds across the 8 minutes or so of the task. So why does this matter? I think I've already kind of emphasized some of this. So one of the things that we're trying to do is say, okay, so here's our little signal. So we take that habituation marker. We actually have been working with folks at Radboud University in the Netherlands. This is Laura Bloch's dissertation work. That's me as a pretend fly scientist. And she has these Drosophila knockdowns where she puts in this little tube and then not listening to sounds, but she looks to see how often these flies jump. And that's related to very specific kind of phenotypes. So we have this delayed group, which is very much like that autistic sticky attention. So, and we're seeing some different gene groups are kind of linked to these different patterns. So the fly genes you see in the bold, and you see MED13L, actually that fly, the SKD fly, has this severe phenotype where they're responsive, but that they stop jumping very, very early on. So even after just a couple of trials. And here's kind of what in this figure I here is what the MED13L group looks like specifically. And so our goal is to say, well, here's what the flies are doing. How does that map onto the humans? And so the idea would be if there's this one-to-one where we see our kids' habituation maps onto how the flies are working, and we can look maybe at some of these different clusters of genes, well, we can use the flies to test drugs really, really quickly. They're cheap, and you've got lots and lots and lots of them. But we can use our kind of social interventions, understanding how our human interventions and therapies are working to help kind of attune that attention brain in our humans, and to hopefully together is really how we get to treatment. So just kind of, I'm just going to throw these all up, and this is the last little bit. So this is kind of where we're headed. So we're looking very closely, particularly after this weekend, at what we're seeing in MED13L. We're going to tell you what we're seeing in these MED13L brains. And then we have, a lot of you have had to answer these puberty questions. That's our big focus point too, is to try to understand, are there maturational shifts that are helping to explain the attention brain? Nico, Dr. Pini's work is, he does a lot of great things to look at this resting state EEG. And so we can actually look to see relative balance in the brain, and how the brain is either tamping down or ramping up activity. And then really long terms, this isn't like next year, this is maybe 5 or more years, we're really hoping to then use these markers in interventions and understand how are these clinical trials working. And then as a plug for where my personal group is going, we do a lot of work together. So one of the things I want to know is, how does your kid's brain respond to you and your brain? So understanding actually that dynamic between kiddos and parents. And I'm definitely over, I'm sorry, but thank you so much for your attention. And
thank you so much. We have time for a very brief Q&A before lunch. So if anyone has any questions.
If no one else. So I didn't quite follow the slide where you showed the correlation between humans and flies. Is your sense that there's real value in using fly models, or limited value for MED13L?
So there is real. I was way too ambitious in trying to even tease that work. So what we have found is that when you pull out the kids that I had at the time with MED13L, I can take every single kids and take their own habituation slope and their reactivity. And that is mapping on to what the flies are doing as a group. I don't necessarily have the fly individual data, although we're working on that. But what's actually really exciting is that that habituation slope is related to other clinical symptoms. So specifically related to some of the adaptive components in the Vineland. And we have a ton of kind of individual difference measures, and I can't remember all of them off the top of my head, but I could pull that data for you.
Yes.
Yeah, I was curious about the roughly 200 published patients and what parts of the world are represented amongst that, and how outreach is to people in other countries.
Yeah, that's a great question. Very relevant, of course. I think that I don't know the exact distribution off the top of my head, obviously, but there is some representation from pretty much most parts of the world. I know there have been really big studies in Japan, obviously Europe, Belgium, some countries over there, obviously us here, even South Asia, Pakistan, a country that we don't really associate with super great medical advances. It's one of my country, but also some advancements there, or some representation from there too. I don't have the exact breakdown. Of course, check our paper out. I kind of, like Dr. Hudac mentioned, I was a little bit ambitious, and I didn't include quite everything that I should have, but our paper will have that. Just check out Frontiers in Neurology in the upcoming few months. But great question.